Hello, I am Rob David from Thermo Fisher Scientific, and this is Double Helix Diaries. In this episode, meet Dr. Ami Patel, a staff scientist at Thermo Fisher Scientific. Today, she'll share her insights and experiences as an emerging scientist, particularly focusing on her embrace of Sanger sequencing. Well, Ami, welcome here to Double Helix Diaries. We are so excited to be talking with you this afternoon.
Thank you so much. I am looking forward to it.
Excellent, excellent. Given our topic today, we thought it'd be great to ask you, as you're a scientist still, in the earlier part of your career, which sequencing methods have you used in school and in your career?
So my journey with sequencing is pretty diverse. I would say I start with my time as a master's student in the United Kingdom. And I was working on using RNAi to knock out genes in Drosophila, or the fruit fly. We were studying the dopamine receptors and overall addiction and the mechanisms involved in there. So for generating those RNAi, we would generate these expression plasmids that were confirmed using Sanger sequencing at the core facility at the university. That was probably one of my first experiences using sequencing. Moving on, I did my PhD here in the Midwest in the US, and my area of research was neurobiology. I was studying neurons that innervate our tongue, or the gustatory neurons, that actually reconnect as adults, which doesn't happen, as we know, with spinal cord injury. So very interesting topic. The way they do it is they use growth factor cues that attract them towards them. And the goal for me was to knock some of these growth factors specifically out and generate mouse models that were mutated for these growth factors. And a routine way of testing whether these growth factors were knocked out was using PCR sequencing regularly during my grad school. From there, for my postdoctoral research, I studied neuro-oncology and these malignant tumors that are super aggressive. And the focus there was to identify the overexpressed genes in these malignant tumors and knock them out. And the way we chose to do that is using CRISPR-Cas9, which was pretty hot back then.
Indeed, yes,
Yeah.
I remember.
I remember using Thermo Fisher's cloud-based software, Seq Screener, gene edit confirmation at that time to confirm, do my data analysis and generate, beautiful images for my publications. And then from then on, right before I joined Thermo Fisher, I worked with a synthetic biology company. And the goal there is to metabolically engineer microbes, like bacteria, fungi, all different microbes, to generate products like polymers, manure, beauty products. And doing that metabolic engineering, we used to generate thousands of strains. And the confirmation for successful strain generation was done using NGS because it was so high throughput at that time.
Wow, so just there, in the short span that we've been talking here, you discussing your journey, we've hit upon cancer research, microbiome, bioproduction, and also spanning the evolution of sequencing itself from Sanger through to NGS.
Yep.
Wow. So that actually kind of makes me wonder, maybe we can do a little bit of word association here. So what is the first word that comes to mind when you hear Sanger sequencing?
The word that comes to mind is the Human Genome Project, because that was the biggest, back in the early 20s when I was still in grad school, the Human Genome Project, that had just completed using the CE-based Sanger sequencing platform on its own. And it was like, wow, like that was, a wow moment for me. So the history of Sanger sequencing dates back way before to, Fred Sanger's invention of the Sanger sequencing by extension and using the dideoxynucleotide terminators. And also, it's just amazing. So I believe that even though NGS has become more of the mainstream methodology, Sanger sequencing will always be like the gold standard reference methodology just for its simplicity of use and the brilliance behind the concept.
I hear you. Yeah, and I think, seminal, when you said, Human Genome Project, that was a seminal moment in our history, in human history, really. But even I remember in my career, that was my first memory of Sanger sequencing too. That was the first technology in molecular biology that I really used as a tech. So super cool. I guess we talked a little bit about history, your experience, and touched cursorily upon these. What about some of the areas of application that you've used with Sanger?
So the one that comes to mind, because I recently have worked with our internal colleagues at Iron Torrent with, is confirmation sequencing of NGS results using Sanger sequencing. So the Oncomine kit is a panel of genes for somatic mutations that are commonly required for detection and diagnosis of several different kinds of tumors. And it covers, I think, about quite a few. I don't know the exact number. But in collaboration with CR&D here at Thermo Fisher, working with Ion Torrent, we have identified or done confirmation sequencing on NGS results using FFPE DNA samples from patient samples, and done confirmation sequencing providing information about deletions, about insertion mutations, about single nucleotide mutations for genes that are commonly analyzed or have mutations like PTEN, TP63, NF1. NF1 actually is one of the genes that I used in my postdoc because it's NPNSD gene.
A nice connection there from both before and after.
Yeah.
Love that.
Yeah.
Yeah, and I just love what you've described there, because you've encapsulated the synergy between capillary electrophoresis and NGS, and that's I think one of the beauties of working with Thermo Fisher.
Yeah, and to add to that, confirmation sequencing is routinely used in clinical labs to do confirmation sequencing for NGS results that are relevant to diagnosis before, as a reflex check for positive results, before sharing them with clinicians or patients.
So we've now our conversation has meandered around around a bit, and I think what we'd like to hear is a bit more of some personally exciting stories, if you can think of, it can be from your past and your research days, or here at Thermo Fisher as well, with capillary electrophoresis technology.
So when I started here about 2 years ago with the CR&D group at Thermo Fisher, the most surprising thing is how versatile the CE technology is. It is so applicable. In that, what I mean is that it allows not just for DNA sequencing analysis, but also fragment analysis applications. The one example that comes to mind is that I mentored a summer intern for the CR&D group this past summer. And her project was revolved around identifying common codon 12 and 13 KRAS mutations. The KRAS, and there's a lot of application for the KRAS mutations because there are this new age drugs are coming out that will target specific KRAS mutations. So it will target the G12C is the most hotspot, and that's the one that they'll target. That's the drug out already. 2 drugs are out there, but there's also clinical trials. So there's a huge, application of this KRAS mutation detection. So her project involved identifying these mutations using DNA sequencing as well as doing fragment analysis. And with somebody who had very minimal experience in the lab, the workflow of doing DNA sequencing or doing fragment analysis was very easily adoptable. So the design was easy and doable. It was a low lift to execute these experiments. The data generated from these experiments, whether it was DNA analysis or it was fragment analysis, was very, very doable for this summer intern to analyze, to put it into slides and present and talk about it. So it was very adaptable to the type of assay we wanted to do. We wanted to go after KRAS mutations. We're going to adapt CE to that. We want high sensitivity assay using the fragment analysis. We were able to design and execute. She was able to design and execute these experiments. Similarly for the DNA sequencing. So the versatility of how you can adopt to the question you're going after is just amazing.
Wow. So you touched on something that's personally I'm passionate about, and that is mentoring young people in
Giving
science. Sorry.
Giving back. Somebody mentored me, and then doing it for another younger scientist who's getting involved in the field.
Exactly. And so often when we're younger and we're starting out, some of the technologies might be intimidating. I mean, we want to dive right in there. But I like how you highlighted that as far as the workflow and even the analysis tools that we have at our disposal, they're approachable, and one can easily start using them and make an impact.
Yeah.
So I've asked you a number of questions here on a few different topics, but often we can miss things. Is there any question that I haven't asked you about concerning capillary electrophoresis, be it sequencing or fragment, that you'd like to share with us today?
I'm still learning, as I'm new to CE technology in terms of being like on the product side and understanding the nitty-gritty of CE technology. But like I said, I gave the KRAS example. The assay development side, we have so many tools on our website that we have primers on our primer designer tool that will do Sanger sequencing or do fragment analysis. They're pre-designed and they cover the entire genome. The breadth of products that we have that support CE technology are just amazing.
They really are. So I think with any aspect in life, and here in science, we often have our favorites. So if you could pick your one favorite attribute of capillary electrophoresis technology, what would it be?
Maybe more than one.
Okay, please.
So flexibility of content, right? Like I said, you can design the assays that you want. You can go after, the biomarker or the mutation that you're going after, and it can be very customized to whatever you want to, what the content you want it to be. The other side is, like I mentioned earlier, it's very flexible for applications. So, it spans from Sanger sequencing to VNTR genotyping to single nucleotide genotype mutation detection to fragment analysis applications, right? And it depends on what you want to do and where you want to go. So that application flexibility is amazing and lets you, do a lot with one technology. And I would say from my experience with the summer intern, that demonstrates the low burden to, use this technology, like the design, the data analysis, you don't need to know bioinformatics to analyze a CE data. And the execution and the design part is very easy, doable, very logical. One of the easiest, I would say, molecular biology techniques to adopt.
What a great way to wrap us up. Customization, flexibility, and approachability. Ami, thank you so much for joining us today on Double Helix Diaries.
Thank you so much. I love sharing all my experiences with CE.